Namaskar and welcome to SYBSc Student friends in this semester number 4. In this semester we will deal with paper number 7 and paper number 8. In this lecture I am going to discuss the unit number first from the paper number 8, that is enzymes and nitrogen metabolism. आता ह्या स्लाइडमध्ये जी समोर दिसते पहिली स्लाइड, त्या स्लाइडमध्ये तुम्हाला काही फनी इमेजेस दिसत आहेत आणि एक एक सरळ असा सोपा प्रश्न दिसतोय. तर एन्झाइम्स जे आहेत हे एन्झाइम्स आर नथिंग बट दी प्रोटीन्स. सो in the funny images you can see what these enzymes can do or what is their function is you are observing in this slide. So as like एक बेसिक प्रश्न असतो की आहे की कोंबडी आधी की अंड आधी. अशाच प्रकारे मॉलिक्युलर लेवलला अणुरेणू स्तरावरती एक महत्वाचा प्रश्न पडतो आपल्याला की which came first, that is whether proteins or DNA or RNA. तर ह्या प्रश्नाच्या अनुषंगानं एक आर्टिकल तुम्हाला त्याचा अभ्यास करावा लागेल. त्याचे लिंक जी आहे तर ती डिस्क्रिप्शन बॉक्समध्ये तुम्हाला मी देईन. सो लेट अस स्टार्ट दी topic enzymes. So in this unit number first, that is enzymes and nitrogen metabolism, what we are going to see that we will discuss in this slide. So in this unit number first, the first subunit, that is enzymes, that we are going to discuss now. That in that we will see total 5 subunits. That is in 1.1, that is we will see the introduction about the enzymes. So in this particular thing, introduction, that means the definition of enzymes, then history regarding or discovery regarding the enzymes, that we will discuss. After that we will see the classification and nomenclature of enzymes, how these particular enzymes get named and what are the different classes of the enzymes that we will discuss. After that, in the third subunit, that is 1.3, in which we will see the how these enzymes look like, that means the structure of the enzymes, then what are the different properties of the enzymes, that we will discuss. After that, in the 1.4, we will see how these enzymes, as the subject is related to the metabolism, we have to understand how these enzymes act or what is the mechanism of these enzyme action. Then the last subunit, that is factors which are affecting enzyme activity. In that, only 2 factors that we are going to discuss. There are many more factors which are responsible for the enzyme activity. In that, temperature and pH, these 2 important factors we will discuss in this unit, that is subunit, that is enzymes. So we will discuss now. Now regarding these enzymes, we will see the introduction. In this introduction, so the first basic point regarding or basic important thing about the enzymes is there are 2 fundamental conditions for the life of the living being. Living entity must be self-replicate. याचा अर्थ की त्या जी काही जीवसृष्टी आहे किंवा एखादा जीव आहे तर त्यानं आपला वंश हा जो आहे हा त्याला वाढवता आला पाहिजे. That means the living entity must be self-replicating or self-replicate. And the second thing, the organism must be able to catalyze chemical reactions efficiently and selectively. So for that purpose, in the biological system, the reactions are catalyzed by special and remarkable proteins, the enzymes. So these are the enझाईम्स जी अशी गोष्ट आहे जी वनस्पतींमधल्या ज्या काही वनस्पतींमधल्या किंवा जीवामधल्या ज्या काही केमिकल रिॲक्शन्स आहेत किंवा ज्या काही चयापचयाच्या प्रक्रिया आहेत तर त्या एफिशियंटली आणि सिलेक्टिव्हली अगदी उत्तमरित्या चालवतात. The enzymes are the most important constituents of biological world. So in Greek, according to the Greek language, the word enzyme is en means in and zyme means east. मराठीमध्ये एन्झाईमला विकर असं म्हटलं जातं किंवा किण्वक असंही म्हटलं जातं. तर हे एन्झाईम हा जो आहे हा ग्रीकमध्ये ह्याचा एन मीन्स इन आणि झाइम मीन्स ईस्ट असा अर्थ आहे. आणि देन दी enzymes, they are the catalyst formed by cell for the specific chemical reactions. So they are also called the biocatalyst. Catalyst means which enhances the rate of reaction. यावरूनच जे काही एन्झाईम्स आहेत तर त्यांना बायोकॅटॅलिस्ट अर्थातच जैव उत्प्रेरक एखाद्या रिॲक्शनचा एखाद्या प्रक्रियेचा केमिकल प्रक्रिये रासायनिक प्रक्रियेचा वेग वाढवणाऱ्या सबस्टन्सला पदार्थाला उत्प्रेरक असं म्हटलं जातं आणि एन्झाईम्स जे आहेत ह्यांना जैव उत्प्रेरक असं सुद्धा म्हटलं जातं. Now we will see the definition of these enzymes. Enzymes are a complex chemical protein or polypeptide produced in a living cell which catalyzes a biochemical reaction at very low concentration. All the enzymes are proteins, but all the proteins are not enzyme. ही गोष्ट फार महत्त्वाची आहे. याच्यासाठी पहिल्या स्लाइड मध्ये आपल्याला जे काही आर्टिकल दिलेला आहे त्यावरून आपल्याला हाही एक नवीन प्रश्न पडू शकतो किंवा ही एक वेगळी गोष्ट समजू शकते की ऑल दी एन्झाईम्स आर प्रोटीन्स बट नॉट ऑल दी प्रोटीन्स आर नॉट दी एन्झाईम्स. The study of enzymes, their nature, mechanism of action has got more practical importance in the life. Many drugs exert their biological effect through enzyme interaction. Enzymes are important practical tools in medicines, chemical industry, food processing and agriculture. So this is the particular scope. हा जो काही एन्झाईम स्टडी आहे एन्झाईम्स बद्दल किंवा विकारांच्या बद्दल आपण सविस्तर अभ्यास केला तर आपल्याला मेडिसिन इंडस्ट्रीमध्ये किंवा केमिकल इंडस्ट्रीमध्ये फूड प्रोसेसिंग मध्ये आणि शेती व्यवसायामध्ये किंवा ॲग्रिकल्चर फील्डमध्ये खूप स्कोप आहे. रिगार्डिंग द डिस्कवरी ऑफ एन्झाईम्स, द बर्थ ऑफ एन्झाईम could be traced with the activity of wine production by the Greek peoples. In 1860, Louis Pasteur first recognized the potentialities of yeast cells to ferment the sugar. Pasteur can be truly recognized as the original detector of enzymatic activity in the living cells. Then afterwards, Frederick Kuhne in 1878 firstly coined the term enzymes to designate any chemical substance generally called ferments. In 1926, J.B. Summer isolated and purified enzyme urease and concluded that enzymes are proteins. Later on, John Northrop and Moses Kunitz crystallized pepsin, trypsin and other digestive enzymes and found that they are also proteins. In 20th century, in 20th century, purification of thousands of enzymes was carried out along with the structure determination, chemical mechanism, and general understanding of mechanism of action. Regarding the classification and nomenclature of the enzymes, how these enzymes get named or how the name is given to these enzymes and what are the different classes of the enzymes that we will see now. So in biological system, many enzymes carry out metabolic processes or many metabolic reactions. In recent years, 2000 different enzymes have been identified which acts on specific substrate class of the substrate. International Union of Biochemistry established International Commission on the Enzymes for systematic arrangement, classification and nomenclature of the enzymes in year 1962 and which is revised in the year 1964. The suffix ASE, आता एन्झाईमला नाव देत असताना कसं दिलं जातं तर त्याच्या शेवटी ASE असं नाव असं सफिक्स लावलं जातं which is added to the name which indicates substrate on which enzyme act. For example, on the urea is hydrolyzed by the enzyme urease. Phosphate splitting enzyme which is called phosphatase. इथं लक्षात घ्या की युरिया जो आहे तर त्याला हायड्रोलाइज करणारा एन्झाइम आहे ते युरिएज. युरिया जो आहे जो सबस्ट्रेट आहे त्याच्या शेवटी ASE असा प्रत्यय लावला जातो. युरिया युरिएज, phosphate splitting phosphatase. The commission recognized 6 major classes which we will see in the next slide also. We will detail discussion regarding this particular 6 major classes will be in the further slides, which further divided into subclasses and subclasses. Now enzymes are recognized by systematic position and code number given by the particular commission. Each enzyme has been given a 4-digit code symbol that we will see in this slide in detail. Nitrate oxidoreductase which is having enzyme code 1.6.6.2. Here EC stands for enzyme code. The first in the detailed 4-digit code, the first digit is that is first, which is the class of enzyme that is oxidoreductase. Then the 6 number, 6वा जो नंबर आहे तर तो आहे 6 नंबर जो आहे दुसरा नंबरचा जो आंकडा आहे तर तो आहे सब क्लास ऑफ द एन्झाइम that is 6 in the 1.6 that is substrate on which enzyme are acting, that is NADH or NADPH as a electron donor. Then the तिसरा आकडा जो आहे तर तो आहे 6. अगेन 6 आहे 1.6.6 that is the हा जो 3 नंबरचा आंकडा आहे तो तो आहे सब class of the enzyme that is 6 that is 1.6.6 आणि हा आहे which transfers the electron to the nitrogen group, and the last code that is 2 which indicates the name of enzyme of the series that is 1.6.6.2 which reduces the nitrate to nitrite. The another examples that is succinic dehydrogenase ह्याचा enzyme code आहे 1.3.9.9. Similarly, the pyruvate kinase ह्याचा enzyme code आहे 2.7.1.40. आता in case of the enzymes are divided into 6 major groups or classes on the basis of the type of reaction which is catalyzed. ज्या एखाद्या रिएक्शनचा रेट एनहान्स करणारा जो काही एन्झाइम आहे और कॅटलाइज करणारा जे एन्झाइम आहे, the type of reaction catalyzed and substrate name are the two factors which provide the basis for naming and the individual enzyme. It also forms the basis for enzyme classification and code number. As per IUB, that is International Union of Biochemistry System, the name of enzyme has two parts like binomial system. जसं की आपल्या प्लांटचं बायनॉमियल असतं तसं हे बायनॉमियल असतं. The first part here at the end of the slide, the first part indicates the substrate while the second part indicates the nature of reaction. Here in the at the end of slide, ATP hexose is the first part while the 6-phosphotransferase is the second part. आता ह्या स्लाइड मध्ये एक टेबल दिलेला आहे, तर त्याच्यामध्ये 6 मेजर क्लासेस ऑफ द एन्झाइम्स आर गिवन. दी 6 मेजर क्लासेस मागील स्लाइड मध्ये आपण बघितलं की एन्झाइम कोड जो आहे तर त्यातला पहिला डिजिट जो आहे, फॉर एक्झाम्पल दी ऑक्सिडोरिडक्टेज हॅव्हिंग एन्झाइम कोड 1.6.6.2, तर ह्याच्यामधला जो पहिल्या क्रमांकाचा आकडा आहे म्हणजेच 1 हा काय दर्शवतो तर क्लास ऑफ द एन्झाइम. अँड दीज आर दी 6 मेजर क्लासेस ऑफ द एन्झाइम. We will see one by one. The table which is having first the number of class, that is serial number which indicates the number of the class among the 6 different major classes. Then the name of that particular class, then type of reaction which is catalyzed by that particular class. Then type of reaction which is catalyzed by that particular class, that particular enzyme. Then we will see the one by one. The first major class is oxidoreductase. Here we can see the 3-dimensional structure of the oxidoreductase enzyme. Then which catalyze the oxidation reaction. Here we can see in the table, I will show you the oxidation reduction reaction which is catalyzed by the oxidoreductase example, oxidoreductase enzyme. The examples are oxidases, dehydrogenases, reductases. Then the second example, that is transferases. Here we can see the 3-dimensional structure of the enzyme transferases. The transfer of functional group is the role of that particular class, that is transferases. The examples are phosphate and methyl group transferring enzymes. Then the third 3 नंबरचा जो क्लास आहे तर तो आहे hydrolysis. Here on the left lower side you can see the hydrolysis 3D structure of the one of the enzyme from the major class hydrolysis, which carries out or which catalyzes the hydrolytic reactions. For example, the enzymes acting on the ester bond peptide linkages. Then the fourth class is the, here you can see the Lyases 3-dimensional structure. They catalyze the addition or loss of double group or double bond. The example is aldolases. The fifth class, that is isomerases. The isomerization type of reaction they carried out. Examples are mutase, epimerase. and the last one, that is ligases. In the first slide you have seen the funny picture regarding this particular ligases, which the role of joining of the हे लायगेजेस जे आहेत हे जोडण्याचं काम करतात. याबद्दल आपण डिटेलमध्ये पुढच्या स्लाइड्समध्ये चर्चा करणारच आहोत. तर सिंथेसिस बाय कंडेन्सेशन ऑफ टू ग्रुप्स रिक्वायर ATP as energy source. The examples: glutamine synthetase, enzyme forming ACOA. These are the 6 major classes of the enzymes, and what type of reactions they carried out. So in the next further slides we will discuss regarding the in detail about these 6 major classes one by one. आता हे जे 6 क्लासेस आहेत एन्झाइम्सचे, तर त्यांच्या संदर्भात आपण सविस्तर चर्चा करूया. म्हणजे डिटेल्ड क्लासिफिकेशन ऑफ एन्झाइम्स इन्व्हॉल्विंग इंटरनॅशनल युनियन ऑफ बायोकेमिस्ट्री रूल्स. सो द फर्स्ट क्लास इज द ऑक्सिडोरिडक्टेज. आता हा ऑक्सिडोरिडक्टेज क्लास काय आहे ते आपण बघू. तर these enzymes catalyze oxidation and reduction reactions. So these main two reactions are occur in this particular class oxidoreductase. A large number of oxidoreductase enzymes are known, where one compound is oxidized and other is reduced after enzyme action. These enzymes bring about the main energy yielding reactions of the living cells. They act by transferring electrons and hydrogen ions. आता याच्यामधली एक्झाम्पल आहे NAD ऑक्सिडोरिडक्टेज. तर याच्यामध्ये अल्कोहोल आणि निकोटिनामाइन ॲडेनोसिन dinucleotide in the presence of NAD ऑक्सिडोरिडक्टेज म्हणजे निकोटिनामाइन ॲडेनोसिन डायन्यूक्लियोटाइड जो काही कंपाऊंड आहे तर तो इन द प्रेझेन्स ऑफ द ऑक्सिडोरिडक्टेज एन्झाइम ह्याच्यामध्ये ऑक्सिडेशन आणि रिडक्शन या दोन्ही रिॲक्शन्स होतात. आणि अल्कोहोल आणि NAD निकोटिनामाइन ॲडेनोसिन डायन्यूक्लियोटाइड यांचं रूपांतर अल्डिहाइड मध्ये होतं आणि NAD जो आहे तर तो इट रिसीव्स द हायड्रोजन atom. Then another example is the lactase dehydrogenase. Similarly, leo-rotatory lactate plus nicotinamide adenosine dinucleotide in the presence of lactate dehydrogenase enzyme converted into pyruvate and the compound nicotinamide adenosine dinucleotide receives the hydrogen. So the oxidation and reduction, both the reactions occur with the help of these oxidoreductase enzymes. The important group of enzymes included in this class are the dehydrogenases, oxidases, hydroxylases, peroxidases, catalases, oxygenases, etc. So this is about the class oxidoreductases. The second class is the transferases. आता हा सेकंड क्लास काय आहे, सायमल्टेनियसली ह्या जे काही क्लास आहेत तर त्या क्लास मधल्या एका एक्झाम्पलचं किंवा एका एन्झाइमचं 3 डायमेन्शनल स्ट्रक्चर जे आहे हे उजव्या बाजूला आपल्याला दिसत आहे. तर हा जो काही ट्रान्सफरेजेस आहेत तर ते ट्रान्सफरेजेस जो क्लास आहे तर तो काय आहे आपण बघूया. These enzymes, so the enzymes of this class are involved in transferring functional group between a donor and an acceptor molecule. म्हणजे ह्याच्यामध्ये काय आहे की या फंक्शनल ग्रुप जो आहे ह्याची देवाणघेवाण ह्या एन्झाईम मार्फत होत असते. These enzymes transfer groups like formyl, glyosyl, methyl, carboxyl, acetyl and phosphate from donor to acceptor. आता याच्यामध्ये ग्लुकोकायनेज एक एक्झाम्पल आहे. याच्यामध्ये ATP plus dextrorotatory glucose in the presence of enzyme glucokinase converted into adenosine diphosphate plus dextrorotatory glucose 6 phosphate group. आता याच्यामध्ये फॉस्फेट ग्रुप जो आहे ॲडिनोसिन ट्रायफॉस्फेट कडून डेक्स्ट्रोरोटेटरी ग्लुकोज कडे ट्रान्सफर झालेला आहे. म्हणजे याच्यामध्ये फॉस्फेट ग्रुप जो आहे वरती दिलेला आहे की ग्लायोसिल, मिथिल, कार्बॉक्सिल, ॲसिटाइल अँड फॉस्फेट ग्रुप्स सच अ ग्रुप फंक्शनल ग्रुप्स आर transferred from donor to the receiver. The second example is glutamate, pyruvate amino transferase, which is the synonym of glutamate. Here the glutamate plus pyruvate with the help in the presence of enzyme glutamate or pyruvate amino transferase, 2-oxyglutarate plus alanine is formed in this particular presence of glutamate. Here similarly the functional group is transferred from donor to the receiver. The important groups of enzymes in this class are aminotransferases, transfer of amino group, kinases, transfer of phosphoryl group from ATP, transaldolases and transketolases, transfer of aldol and ketol group. So this is about the second class, that is transferases. The third class, that is hydrolases. आता ह्या तिसऱ्या क्लास मध्ये जे काही एन्झाइम्स आहेत ते कशा प्रकारे वर्क करतात ते आपण बघू. These enzymes are special kind of transferases. म्हणजे similarly the second class transferases, these are special kind of transferases where the donor group is transferred to water molecule. The enzymatic reaction is that of hydrolytic cleavage. म्हणजे हायड्रोलायसिस म्हणजे काय की ब्रेकडाऊन जे आहे तर ते ह्याच्यामध्ये होतं. These enzymes split carbon oxygen, carbon nitrogen, carbon carbon, oxygen phosphorus and carbon sulfur bonds. The examples, आता याच्यामध्ये एक्झाम्पल जे आहेत तर ते ATPases आणि लायपेजेस जे आहेत ते आपण बघू. तर ह्याच्यामध्ये ATPase जे आहे एन्झाइम which in this particular reaction ATP plus water molecule in the presence of ATPase converted into adenosine diphosphate plus inorganic phosphate. That means the special kind of transferases where the donor group is transferred to the water molecule. Here in this the donor group transferred to the water molecule and the products you can see the adenosine triphosphate plus inorganic phosphate. Similarly in case of the lipases, fat plus lipids or fats plus water molecule in the presence of lipase converted into fatty acid plus glycerol. Similarly the donor group is transferred to the water molecule. The important types of hydrolases are lipases, peptidases, phosphatases, amidases, deaminases, glycosidases and ribonucleases. This is all about the third class, that is hydrolases. The fourth class is the lyases. In this particular enzymes of this group catalyze the addition of group to double bonds or catalyze cleavage by electronic rearrangement of molecules. Lyases work in these two ways. These enzymes act on carbon-carbon, carbon-oxygen, carbon-nitrogen, carbon-sulfur and carbon halide bonds. The example First is the decarboxylase, which removes the carbon dioxide from keto acid. Fructose diphosphate aldolases. Here in the presence of this enzyme fructose diphosphate aldolases, fructose 1,6 diphosphate is converted into glyceraldehyde 3-phosphate and dihydroxy acetone phosphate. This group includes decarboxylases, aldolases, etc. So this is about the fourth class, that is lyases. The fifth class is the isomerases. These particular enzymes catalyze the process of isomerization. That means the similar structure of compound is formed with the help of this particular isomerases. These enzymes catalyze isomerization reactions of several types. There is a redistribution of chemical atoms within the molecule. Due to isomerization reaction, there is intramolecular rearrangement of atoms in substrate. They include cis, trans, keto, enol and aldose ketose interconvergence. म्हणजे एक प्रकारे एका कंपाऊंडचं सिमिलर केमिकल फॉर्म्युला असणाऱ्या दुसऱ्या कंपाऊंड मध्ये रूपांतर होणं. ह्याच्यामध्ये इपिमरेज जे एन्झाइम्स आहेत, एक्झाम्पल दॅट इज दी डिझायल्युलोजेस 5-phosphate in the presence of epimerases converted into D-ribulose 5-phosphate, which is of course it is the reversible reaction. The triose phosphate isomerases, the dextrorotatory glyceraldehyde 3-phosphate in the presence of these trioses converted into dihydroxy acetone phosphate. This group includes epimerases and some mutases. So this is about the isomerases, fifth class isomerases. The last class, that is ligases or synthetases. Here in the funny picture you can see that the enzyme ligases, which is saying that just making the ends to meet. Especially in the DNA polymerization, this particular enzyme is helpful in the joining of the bonds or joining of the polynucleotides. So let us see about these ligases. These enzymes catalyze bonds formation and are associated with hydrolysis of ATP or other nucleoside phosphate. They are also called synthetases. The example is glutamine synthetase, and the reaction regarding this particular glutamine synthetase, in which the glutamate plus ammonia plus the ATP molecule in the presence of glutamine synthetase converted into glutamine plus hydrolysis of ATP molecule occurs and water molecule is formed. Regarding the remaining part of the topic enzymes, that is the structure and properties of enzymes, mechanism of enzyme action, how this particular enzyme works, then different factors, that is pH and temperature, what is the effect of these factors on these enzymes, and the enzyme inhibition. These particular things we will discuss in the second part of this particular enzymes. So thank you.